After studying this module, you shall be able to know about the various body fluids. You will able to know the forensic aspect of these body fluids as a evidence. You will also able to examine all these body fluids. Forensic biology and serology is a branch of forensic science which deals with the biological evidence and their examination. In the examination of crime such as the murder, robbery, rape, etc., the examination of biological materials play an important role in connecting the crime and the criminals. Such biological specimen may be in the form of body fluid, stains or materials like the blood, semen, saliva, urine, fecal matter, milk, hair. The most important evidences of body fluids are blood and semen from personal identification point of view. Blood. First, let us know the composition of blood. Blood is a complex viscous red fluid with a pH of about 7.4. It is mainly composed of two parts, that is cells and the plasma. The liquid part is called plasma and the solid are called red blood cells, which is also known as erythrocytes. Then we have the white blood cell, leukocytes and the thromboblasts, that is the platelets. The cells are also known as corpuscles. When the blood flows out of the body, a part separates as a blood clot consisting of blood cells, disc and fibrin. Fibrin comes out of the plasma and is responsible for clotting of blood. The liquid left is called serum. It is the plasma without fibrinogen which turns into fibrin. The red cells. There are about 5 million red cells per microliter of the blood, each about 10 micron in diameter. The red cells are continuously being produced and destroyed in the body. The rate of destruction is about 10 billion cells per hour in a normal healthy adult. The hemoglobin. The coloring substance of red blood cell is hemoglobin. The substance is of great importance to the body function as it is the carrier of oxygen, injected medicines, salts and foods to various body tissues. It is useful in blood examination. White blood cells. The number of white blood cells is about 4 to 11,000 cells per microliter. They resist the attack of diseases. Platelets. The number of platelets is about 5 times that of the white blood cells. They facilitate the blood clotting. Serum. Plasma and serum are complex mixture of proteins, minerals and organic compound dissolved in water. The functions of blood. The blood performs various functions, that is, it carries oxygen and food to body tissues. It helps to eliminate waste matter or products of the body tissue through kidney and other parts of the body. It regulates the temperature and acidity of the body. It carries administered medicines to the affected parts of the body. Examination of blood evidence. The identification and evaluation of blood is based in its composition and behavior under various conditions. First is cell structure. Since blood is composed of red and white cells, platelets and plasma, the cells has defined shape and size which is different species. For example, human cells have characteristic non-nucleated disc with a diameter of about 0.08 mm. The identification of cell structure is possible when the blood is fresh and moist, usually less than 1 day old. When the blood has dried, it is not always possible to completely regenerate the blood cells. The study of cells is made through microscopy. Enzymatic activity. Blood contains heme group in hemoglobin. It behaves like peroxidase. It catalyzes the liberation of oxygen from oxygen-rich compounds like hydrogen peroxide or sodium perborate. The oxygen is used to carry out certain color reactions. The common color reactions for blood detection are benzidine, phenolphthalein, leucomalachite green and luminol reaction. Other enzymes have been utilized in electrophoretic techniques when they are subjected to electric field due to some polarity, they move towards the opposite polarity. The rate of movement varies with their structure, weight and electrical charge. The pattern of separation is characteristic of blood. Spectrophotometry. Hemoglobin on treatment with acids or alkaline producing agents or oxidizing agents gives a variety of products which have characteristic absorption spectra. They help to identify the blood. The absorption bands are seen in length which are characteristics of blood. Visual examination. It is useful to determine whether the given stain is of blood or not. Ordinarily, there is no difficulty in fresh but old stain may not be identifiable. The examination permits the determination of number and size of stains, rough estimate of amount of blood shed, the direction from which the blood has fallen is found from the tip of the elongated stain. The tip of elongated points out to the direction. The shape of the stain indicates the height from which the blood has fallen. Like the blood stain from the height up to 50 centimeters are round sharp edges. Blood stains from the height of 50 to 150 centimeters have small spike like projections along its edges. Blood stain fallen from a height over 150 centimeters have corrugated edges. From the position of the stains, the movement of the victim and the culprit can be determined. If the culprit run away bleeding, he can be tracked through blood stain. The degree of fluidity, dryness and changes in the color permits rough estimation of the age of stain. The gross foreign materials like hair, flesh, bones in blood may be identified the site of injury. The ultraviolet and infrared examination. Ultraviolet or infrared rays reveal washed or invisible blood stain from the clothes, furnitures, earth, doors, etc. They also reveal the stains on the colored garments or on the painted surface. They help to reveal minute blood traces. Microscopy. Microscopic examination of blood stain is important in many situations, like the species of origin of fresh blood stain can be established through microscopic studies and micromeasurement sometimes. The part of body from which the blood has from the nature of extratenacious matter. Thus, blood from nose, mouth, vagina or anus can be identified. Diseases like leukemia or syphilis can be detected microscopically. Menstrual blood can be identified. Blood from an infected site can be identified through the presence of pus. Spectroscopy. Spectroscopy examination of blood is very useful as well as convenient. The test is usually carried out microscopically. The blood hemoglobin is changed in 2 or 3 on the slide itself and characteristic absorption are observed. Usually alkali hematine and cyanohemogen are studied for their characteristic spectra. Chromatography. The ascending paper chromatography using acetic acid, methanol and water solvent system has been employed to study the RF values. Electrophoresis applied to the blood stain. It is useful for separation of various enzyme systems. It is being adopted to study the body proteins. This technique is becoming important to distinguish between the blood samples. The color test. These tests are the first series of tests employed after visual study of stains. If a stain gives positive color reaction in any of the 2 color tests, the stain is possibly a blood stain. If it fails to give a color reaction, In all probability, it is not a blood stain or same cannot be established. The benzidine reaction. Benzidine, that is 0.1 gram, and dry sodium perborate 0.1 gram are dissolved in glacial acetic acid 10 ml and spread over the stain. A strong blue color indicates the blood. Leucomalachite green reaction. The reaction is prepared by dissolving leucomalachite green 0.1 gram in sodium perborate 0.3 gram in 65% of glacial acetic acid. The reagent is applied to the stain. Intense green color indicates the blood. Phenolphthalein reaction. Phenolphthalein 1 gram is reduced and dissolved in acetic acid, that is 100 ml. Sodium perborate 1.4 gram is dissolved in solution and applied to the blood stain. Pink color indications blood. Luminol test. Luminol is a chemical which has been used to locate the blood stain. The article suspected to bear the stain is sprayed with the luminol. It reacts with the blood to give fluorescence. The blood are made visible. Even decomposed blood reacts with reagent. The reagent does not interfere with the subsequent blood test. It is prepared by dissolving sodium perborate, sodium perborate 0.7 gram in water 100 ml and adding 3-arenophenolphthalein hydrazine, that is 0.1 gram, and sodium carbonate 5 gram to the solution. The solution is spread upon the article with the glass sprayers in the dark room. Bloods give strong luminance. The crystal test. 2 crystal tests are commonly employed. They are specific for blood, but they are not sensitive. They often fail if the conditions are not rigidly controlled or if the blood is disintegrated or contaminated. The Teachman test. Take a dry crust or smear of blood on a slide. Put a drop of potassium iodide, bromide or chloride, that is 0.1 gram, solution in 100 ml of glacial acetic acid over the blood and cover it with cover slip. Warm the slide gently till it gives out bubbles. Typically, Semen crystals are observed under microscope. Heating may have to be repeated number of times. Takayama test. It is also performed similarly. The reagent is prepared from 1 volume of glucose solution, that is 10%, 1 volume of potassium hydroxide solution, that is 10%, and 2 volumes of pyridine, which are dissolved in a 6th volume of water. Precipitation reaction. The reaction is performed to identify the species of origin of the blood stain. It is very delicate test and requires only small amounts of blood. A dilute blood solution is used for precipitation reaction. The antisera are not diluted. The blood solution and antisera should be free from turgidity and contamination. Semen. Nature of semen. Semen is found in a liquid form, smears or stains, or it may be found in the vagina, anus or rectum. Fresh semen is a gel-like fluid which liquefies on exposure to atmosphere. A normal ejaculation is about 3.5 ml, containing about 30 million sperms. The dry weight is about 7% of the liquid weight. The sperm has definite morphological structure. Its identification in a stains establishes the presence of semen. The shape and size of human spermatozoa is characteristic, but the morphology alone does not permit individualization. Semen of a person does not contain any spermatozoon, then it is also called aspermic semen. This is maybe due to some disease. It may be due to vasectomy operations. In such cases, the criteria for identification of semen is lost. Immunological tests using anti-semen smear against the seminal plasma are increasingly accepted as reliable tests for aspermic semen. Electrophoresis is becoming popular for identification of semen. Composition of semen. It is composed of a mixture of organic and inorganic compounds. Important constituent of semen from identification point of view are the proteins including enzymes, blood group factors, choline, fructose, citric acid, uric acid and zinc. The composition varies from individual to individual. Enzymes, acid phosphatase found in the semen is in the concentration which are significantly higher than those found in other body fluids. Acid phosphatase offers a very delicate test for the identification and location of semen stain through a positive identification of semen are not based upon the acid phosphatase test alone. Choline is a semen is used to get crystal test. Fructose, citric acid and zinc are more or less absent in the body fluids and hence their detection in semen should permit its identification, but these substances have not been utilized to any appreciable extent so far. The saliva. Saliva stains may be found at the seam or handkerchief, on discarded cigarette stubs, spittle on cups, tumbles, bottles, postage stamps or envelopes or even the toothpicks. They may be found on the piece of cloth used as gag. Saliva contain an enzyme which when added to starch hydrolyzes it. Saliva extract therefore when added to starch inhibits its color reaction with iodine. Saliva of secretors contain blood group substances and can be grouped. Saliva on cigarette stubs is often in criminal investigation. DNA profiling of saliva stain has enhanced the evidential value of the saliva stain and has brought it at the par with the other body fluids like that of the blood and semen. Food material mixed in saliva may interfere with the blood grouping. Saliva does not give specific precipitation test. Urine. Urine is identified from comparatively large amount of urea in it. The stain is located in ultraviolet rays. It is then extracted with the water and tested for urea. Urine stain can now be individualized through DNA profiling. Feces. Feces are involved in cases of sodomy and bestiality. These stains are found on the garments, on the flies of the trousers and on the flaps of the t-shirts of the active agent. The feces are identified microscopically. Bacterial study of the feces may also lead towards individualization, but the same has not been exploited as a routine at present. It is possible to identify human or animal origin of the feces. Sweat. Sweat stains are involved only in the cases when some garments are left at the scene of crime or where culprit had perspired profusely and left sweat stains. The composition of sweat is similar to the urine. However, if two substances are warm, the odor is characteristically different. Paper and gas chromatography techniques hold bright future for identification of sweat stain. Sweat stains of secretors can be blood group, DNA profiling of the stains as possible. Nasal secretion. Nasal secretion is similar to the chemical composition to saliva. They can be blood grouped if they are belonging to secretors. They are liable of DNA profiling. Tears and human milk. Tears and human milk stain likewise permits the identification. Blood grouping of the same is possible if they have came from the secretors. DNA profile can be developed for individualization. Hair is one of the common and important physical evidence encountered in a crime scene. Individualization of human hair, that is, whether it is from a head or a body, forcible removal of hair may have the blood or skin with the root. In such cases, DNA typing can be done. There have been advancement in the DNA typing technology. Earlier, only hair strands with the root attached could be used for typing. But now, with advanced technology, hair without root can also be used if there are enough number of hair strands. Identification and comparison of hair. Examination of hair evidence found at a crime scene is the first step which should be performed. Hair should be examined to establish the origin of the hair, that is, whether it is a human hair or animal hair. Comparison of hair with victims and suspect should be done in case of human hair. Differentiation of human and animal hair can be done with the ease. Human hair comparison should be done with the extreme caution. variable characteristics can be exhibited by hair not only from individual to another but also within a single person. Morphological features of hair can be observed by careful microscopic examination. These features help in differentiating between human hair and animal hair. Control sample should always be considered before reaching to a conclusion. The main characteristics that should examine are medullary index, scale structure, medullary shape. As forensic evidence, hair is used to compare and determine whether the hair recovered crime is of same origin as the suspect or not. Scalp or pubic hair are most common found at crime scene. The color, length and the diameter of hair are the most important characteristics that should be matched while comparing hair samples. Presence or absence of medulla, its distribution, shape and color intensity of the pigment granules found in the cortex are also important characteristics for comparison of hair. Hair grows at an average of 1 cm per month. There are certain features which are observed and compared for the identification of hair, like color of hair, area of origin, presence of micro-residual material, unusual appearance of hair and the pigments. Forensic biology and serology is a branch of forensic science which deals with the biological evidence and their examination. Blood is a complex viscous red fluid with a pH of about 7.4. The coloring substance of red blood cell is hemoglobin. The benzodine, that is 0.1 gram, and dry sodium perborate 0.1 gram are dissolved in glacial acetic acid 10 ml are spread over the stain. A strong blue color indicates the blood. Luminol is a chemical which has been used to locate the blood stain. Semen is found in a liquid form. Semers or stains, or it may be found in vagina, anus or rectum. Semen is a complex mixture of organic and inorganic compounds. A semen stain, if fresh, is colorless or pale yellow. It gives characteristic smell. A small amount of semen is treated with the iodine in potassium iodide solution. Characteristic crystal of choline iodide are formed. Saliva or secretors contains blood group substance can be grouped. Urine stain can now be individualized through DNA profiling. Feces are identified microscopically. Density is one of the most important physical properties of hair. The reflective index of hair is determined by backline method.